Okay, so today we're going to be talking about immunological tests. So this is by me, Alyssa, Diana, Elise, and Mara. Next.
So first we're going to talk about the Precipitation test. Next.
Okay, so the history of the Precipitation test. In 1897, Rudolf Krauss first introduced the fluid precipitin test, and then in 1901, Paul Uhlenhuth discovered how to make precipitin by mixing chicken egg protein injected in a rabbit with egg white. George Natal discovered that groups of related animals can produce different amounts of precipitin in rabbit anti-serum. Then John Glaser developed the precipitin test to identify mammalian blood. He published this in 1915 in one of his books. Tests first used to support Glaser's evidence Oscar Slater's trial in 1908, and by the 1930s, courts were accepting the precipitin test as a standard practice. John Glaser Jr., the son, obviously, of John Glaser, made the test more accurate and more precise in 1926. Next.
Okay, so what is it? Basically, the Precipitation test is used to detect antibodies or antigens, and it works because the antibodies or the precipitants react with antigens. These antigens diffuse towards each other, and then they form a visible precipitate. It results in an antigen-antibody complex, which are visibly opaque, and they form about minutes to hours after the test. Next. So currently, the precipitin test is used heavily in forensics and in serological testing. It can be used to prevent bioterrorism. So the military research developed a rapid handheld immunochromatographic device, and it detects the presence of B. anthracus protective antigen, otherwise known as anthrax. It's also used in many forensic laboratories and can be used directly at the crime scene. Those are the only companies that I was able to find, but it is heavily used by many forensic laboratories. Next.
Can it be used for COVID testing? The purpose of the precipitin test is to determine the species of blood evidence. It could be, but there are more reliable methods. This test determined blood evidence species, while COVID-19 test should be testing for viral proteins and antibodies.
Okay, so agglutination assays.
So for the history, there were originally two scientists, Herbert Durham and Max von Gerber, which discovered specific agglutination in 1896. And then later on, a French physician used it to test for typhoid fever in 1897. Then we have Rebecca Lansfield alongside her colleagues who used agglutination tests to check for the strep bacteria, which was developed in the 1920s. Next slide.
Okay, so what is it? Agglutination tests are tests used to detect the presence of specific antibodies in serology labs and blood banks. So how it works is latex beads are coated with a specific antigen they want to test. It's then mixed with the patient's immune serum. If the antibodies for the specific antigen is there, they'll react and bind to the antigen, which will cause the agglutination. And this reaction indicates the presence of the antibody that they wanted to look for.
So how is it used currently? Agglutination tests are used to diagnose diseases that are difficult to detect due to whether the organism is difficult to be grown in a laboratory or the disease itself can't be grown. So you have direct agglutination, which is used to detect different strains of the same series. And then you have indirect agglutination, which involves antibodies being latched to latex beads, and it's used to detect variations in bacteria and viruses. Hemagglutination is similar to other agglutination tests, except the antigens are on the surface of the blood cells, so it's used for blood typing. And companies that use it, this was a bit difficult to find, but companies such as LabCorp use it when they're doing blood typing. Next slide. Can it be used for COVID testing? Like the previous tests, technically it could be. However, no serology serology tests are available for COVID-19, and the CDC uses and currently recommends the PCR method, which is polymerase chain reactions, and this is used to diagnose infection based on the presence of a viral RNA. The two potential serology tests for COVID-19 would be one, a test for viral proteins like a Western blot with antibodies raised in animals that would allow for detection, or more commonly, number two, an ELISA test that detects the patient's antibodies to the virus.
Okay, thank you.